Wilder Penfield was a neurosurgeon that was primarily active in the middle of the last century. And he did a lot to pioneer the technique of stimulating small areas of the brain electrically while the patient was awake, with the brain exposed and a portion of the skull removed. Now, the reason he did this was because he was working with patients who had intractable epilepsy, and by stimulating small areas of the brain, he could get a sense of what that part of the brain did for the patient by seeing how the patient reacted to the stimulation. And he cataloged all of it in his 1954 book that he wrote with Herbert Jasper, entitled Epilepsy and the Functional Anatomy of the Human Brain. Now, this landmark book was, to my knowledge, the first kind of whole brain view of how the brain works and what the functional anatomy of the brain really is. In this video, though, I want to try and zoom in on one really interesting finding that Penfield shares. So here's a quote from the book. "Stimulation of the anterior portion of the cingulate gyrus causes patients to stop breathing. However, if the patient is called upon to breathe voluntarily during apnea thus induced, he is able to do so. This is quite different from the irresistible effect produced during apnea induced by Rolandic stimulation." So, what does this tell us? It tells us a couple of things. One, stimulation of the anterior cingulate cortex can stop patients from breathing, which is weird because the anterior cingulate is an area of the brain that usually gets thought of as being important for processing errors or sensing reward value, not really something to do with breathing. And then the other thing that it shows us is there are multiple mechanisms that are involved in controlling breathing. We can see that the mechanism that controls breathing and is somehow getting stimulated when the anterior cingulate is stimulated is one that can be overridden by conscious volition to want to breathe. Whereas when an area of the motor cortex along the Rolandic sulcus is stimulated, there's no possibility of overcoming it. That's just gonna happen. So consciousness can't overcome stimulation in that area. Recently, I've kind of been digging back into it, and what does this really show us? And what I found was there's this really interesting finding from Berger-Cotta in 1951, a couple years before Penfield published his book, and this was actually part of what inspired Penfield, because what Cotta had discovered in looking at data from anesthetized monkeys was that when he stimulated parts of the brain in the anesthetized monkey in a similar way to the way that Penfield was stimulating parts of the human brain, he found that there was actually much more than just the anterior cingulate that seemed to control breathing. There was this big band of respiratory arrest that ran all the way from the medial temporal lobe down on the ventral side of the brain up to the dorsal cingulate on the dorsal side of the brain. So how can this be? Obviously, not all of these areas control breathing. The brain's got other stuff to do. Hi there. I'm Adam Dede, and welcome to a Neuroscience Journal Club. Now, this week's going to be a little different. I'm going to talk about this old finding, the 70-year-old finding, and try and see if by integrating things that have been found since then, we can make a little bit more sense of this wall of respiratory inhibition that seems to run all the way across the whole brain. So, first we got to realize, of course, definitely not all these areas are involved directly in respiration. And so what does this show us? This is a weird piece of data. One reason that this finding feels so crazy is we're thinking about it primarily in terms of respiration. We're not thinking about that arrest, or maybe rephrase it, inhibition part of what's going on here. So if we rephrase and we think about this as a wall of inhibition rather than a wall of respiratory control, then can we see other examples along this band of cortex that seem to indicate that this whole area is somehow involved in inhibition? And it turns out, yeah, we can. So I'm going to talk about two areas. I'm going to focus in first on the anterior cingulate cortex, that area that Penfield was talking about. And so back in 2001, Todd Braver and his colleagues published a paper in which they had participants doing a simple go, no-go task. So in this task, letters would be shown one at a time on the computer screen. And the participant's task was to press the space bar every time a letter was shown, but with one exception. If the letter X was shown, then participants were supposed to do nothing. They were supposed to not go or withhold their pressing of the space bar. And it turns out that this is actually a fairly difficult thing to do. If you're constantly pressing the space bar every time a letter pops up, it's going to be pretty hard to inhibit your prepotent response to want to press that space bar again. And it turned out that the anterior cingulate cortex showed really pretty strong activation whenever the participants had to inhibit a response. So this was evidence then that the anterior cingulate cortex is important for inhibiting unnecessary or pre-planned or non-conscious responses or behaviors. Since then, it's turned out that the anterior cingulate is actually responsible for even more inhibitory signaling. So in another paper, Matthew Weaver and his colleagues, they were having participants do two different tasks. So in one task, the participants were trying to pay attention to a particular stimulus on the screen while ignoring another stimulus on the screen. And they manipulated how salient that distraction stimulus was, so how much it popped out or grabbed attention. And it turned out that the participants who showed a stronger inhibitory signal in the anterior cingulate to that distracting stimulus were the same participants who in a separate task had better working memory capacity. And so here in this figure, what we're seeing is that inhibitory signal in the anterior cingulate to distraction. And on the top, you're looking at participants who had strong inhibitory signaling and had strong working memory performance. And in the bottom, you're looking at participants who had weak inhibitory signaling and poor working memory performance. And just to orient you to the graphs a little bit more, each circle is kind of like a top-down view on a head, with the little triangle on the top of each one indicating the orientation of the nose of the participant. And more dark blue colors indicate stronger signal. And in other studies, it's been shown that this EEG or electroencephalogram measured signal that we're looking at here is in fact related or caused by the anterior cingulate cortex. So this is pretty wild now, right? Like, we can integrate these different findings and see that the anterior cingulate cortex, back from the Penfield and the Kata findings, is responsible for breathing inhibition in some way, but then it's also responsible for inhibition of ongoing or automatic actions. That's what Todd Braver showed us. And then Matthew Weaver is showing us that it's also responsible for inhibiting distracting stimuli, or maybe even distracting thoughts. So it has all these different things, but they're all tied together by inhibition, and specifically inhibition of kind of ongoing or automatic activities. Okay, so what about the rest of this wall of inhibitory, respiratory inhibition? Are there other examples of inhibition going on this wall? And yeah, there are. So next, I want to turn to the rhinal cortex. This is an area in the medial temporal lobe down on the bottom of this band. And back in 2004, Marco de Curtis and Dennis Pare, they actually published a paper where they thought there was so much evidence that the rhinal cortices are inhibitory that they titled the paper, "The Rhinal Cortices, a Wall of Inhibition Between Neocortex and Hippocampus." And they present a whole bunch of different findings showing us that, yeah, there's actually this really strong inhibitory block between the cortex on one side of the rhinal sulcus and the other. And just to give you another picture of where it is we're talking about, here we're looking at a coronal view through a monkey brain, so kind of a head-forward view through a monkey brain. And the rhinal sulcus is that sulcus kind of down on the bottom there, and it separates the cortices that lead into the hippocampus, that kind of swirling structure, from the rest of the temporal lobe. Now, de Curtis and Pare, they present a whole bunch of information, so I'm just going to zoom in on one particular finding, which resulted from data collected from guinea pig brains. Now, it turns out you can take a guinea pig brain out of the guinea pig, and as long as you keep it warm and in the right type of saline solution, it'll stay alive for a while, and you can do experiments with it. So here we're looking at a view of a guinea pig brain from the bottom, and we're going to kind of zoom into the rhinal sulcus and the surrounding rhinal cortices. And in this figure, you can see areas 1 and 2 are separated from area 3 by that line, which represents the rhinal sulcus. So the experiment was to stimulate electrically the olfactory bulb. And as you may know, olfaction or smell has a particularly strong connection with memory. It turns out that the neural signals for smell arrive in the memory centers in the medial temporal lobe far more directly than do the signals from any other sense. So, unsurprisingly, the stimulation in the olfactory bulb showed up in the rhinal cortices very quickly. Importantly, what we're looking at here is the reaction in those rhinal cortices of the olfactory stimulation. And what you can see is that there's a whole bunch of activity going on in areas 1 and 2 on one side of the rhinal sulcus, but on the other side of the rhinal sulcus, there's nothing. The activity does not spread across the sulcus. And this was the theme of a lot of the work that de Curtis and Pare presented in their paper. Every time activity would happen on one side of the sulcus, it would not propagate to the other, or at least it would propagate with a much, much lower probability. So what's the purpose of this? Well, it stops memory from being overloaded. Not everything you encounter is important. Not everything you encounter needs to be remembered. And by inhibiting certain information from making it through into the hippocampus, you can inhibit the non-important stuff from making it through and getting encoded. Okay, so let's zoom back out. So we've seen then that the anterior cingulate cortex is important for inhibiting ongoing or automatic processes. The rhinal sulcus is important for inhibiting information from making it into memory. And both of these, along with the whole area of this band, are important for inhibiting respiratory activity, at least in an anesthetized monkey. So how can we tie this all together? So first, I think it's important to keep in mind that electrical stimulation is not natural activity. So we have to keep that grain of salt in mind. But then second, I think the big thing these data show us is that the differences between respiration, ongoing activity, and memory might not actually be as big as we generally think they are. We think these differences are really big because our folk psychology, our general intuition about the way the mind works, groups things into memory, current perception, attention, so forth. But the brain may not actually care about those constructs that we've come up with. And Buzaki, in his 2006 book, Rhythms of the Brain, makes this point that every field of science only really gains independence as its own field when it starts using its own terminology and dispenses with folk terminology. So when we think about these seemingly disparate findings, what they might really be showing us is that the brain has this strong inhibitory signaling system that separates top-down control in the prefrontal cortex from bottom-up control in the sensory cortices down below. And it doesn't really matter what type of information is being passed between these areas. There's this inhibitory wall that runs through the whole brain, separating them and unifying these different findings. So when Penfield and Kata were stimulating these areas of the brain, what they didn't realize they were doing was actually queuing up activity in a generalized inhibitory signaling system. And that generalized inhibitory signaling system was just inhibiting all kinds of activity broadly, where respiration was just one among many. Anyway, I hope you found this interesting. Please jump down to the comments section below if there's anything that you want to share, any interpretation that you have or disagreement, what have you. If you like the content, please consider supporting me on Patreon. And that's it for now, so catch you next time. All right, bye.